name's Rebekka Koeck. I'm a bioinformatician at the Medical Microbiology Department of the Maastricht University Medical Center in the south of the Netherlands. My poster today is about our work looking at plasmid-mediated antimicrobial resistance gene transmission between different host domains. We're interested in this because farm animals are a potential reservoir of antimicrobial resistance genes, which, if they spread to humans, could cause a problem for the treatment of infections and also reduce the efficacy of our infection prevention measures. To look at this, whether there is a transmission between the different domains, we're involved in the I-for-One Health project. This is a project at the Dutch-Belgian border region where samples were taken from humans in different settings, so hospital patients, care home residents, and also healthy individuals, and also farmed pigs and chickens. The initial results of our study showed that there was little evidence for clonal transmission between these groups, so we were actually wondering whether there was maybe more evidence for plasmid-mediated transmission between the different domains. To look at this in more detail, we've supplemented our Illumina sequencing data with Oxford Nanopore long-read data, and we achieved sufficient coverage in our data for 355 isolates. We then used the MOB suite and plasmid similarity tools to characterize the plasmids in more detail. And these are the results that I'm showing today. One of the first things we noticed is that in the chicken isolates, there were a higher number, on average, of AMR gene-carrying plasmids than in the human isolates, which are shown in blue. We then used the MOB suite tool to classify the plasmids into different groups based on their backbones. And here you can see that we found 14 different backbone classes that were present in at least 2 of the domains. The largest of these is the AA474 plasmid backbone, which we found in 62 isolates from all 3 domains, so chickens, humans, and pigs. However, the MOB suite tool only classifies these based on their backbones and doesn't take into account the cargo that the plasmids may have. So for this, to look at specifically the cargo, so the AMR genes, and also to calculate a similarity measure between the genetic sequences of the plasmids, we used the plasmid similarity tools. Here I'm again showing the results from the AA474 group and all 62 isolates, and you can see that the AMR gene cargo is actually quite diverse amongst the group. And if we implement a Jacquard dissimilarity threshold of 0.25, shown by the red line, we actually find 15 subclusters in this one AA474 group. Of the 15 subclusters, there are 2 which span multiple domains. So overall, in this study, we have found some evidence of potential transmission of plasmids between different host domains. And this just goes to highlight that we do need to investigate plasmids both in humans and potential reservoirs to look for potential transmission events and also to inform our infection prevention strategies. I'd like to thank our collaborators from the I-for-One Health project and the Zonnebay and Interreg funding for making this project possible. Thank you.